Hello students, in the previous session we saw about the cell organelles of plants. In this session we will be seeing the cell organelles of animals. Now if you have a look at the diagram of animal cell, the animal cell will not have a cell wall. It will have only the plasma membrane or the cell membrane. So the outer covering of an animal cell is the cell membrane or the plasma membrane. Now we all know the plasma membrane in eukaryotic cell is made up of lipid bilayer, that is the phospholipid bilayer, which will have a hydrophilic head and a hydrophobic tail with integrated proteins and peripheral proteins. This we have already studied in the last session. Also along with that there are a few differences. Now if you just have a look at the diagram of animal cell. In the animal cell there is one organelle which is added up, which is the lysosomes. So lysosomes, this is the organelle which is having a single membrane. These lysosomes are present only and only in the animal cells. The main function of this lysosome is that it is called as the suicide bag of the cell. Why suicide bag? If the lysosome feels that the cell is in danger, it will release all its contents. Its contents are all different types of destructive chemicals. It is said that this lysosome contains 40 different type of chemicals. 40 different type of chemicals. Now these chemicals which it will release are responsible for breaking the cell membrane and allowing all the contents of the cell membrane outside. Means whatever is present inside the cell, that is the cytoplasm, all its cell organelles will come out of the cell if this lysosome will go out and leak all its contents. And that is the reason it is called as the suicide bag. This is not the only function of lysosome. Lysosome is also involved in killing off any foreign particle which comes in. So if the cell is invaded by a foreign particle, lysosome will be the first one to go and release its enzymes and kill that foreign particle. Also, when a cell will starve, during starvation, lysosomes will go near the food granule, release its content and take the food and derive the nutrition from it. Now let's have a look at the Golgi apparatus. Same way what we have studied in plant cell, Golgi apparatus are derived from endoplasmic reticulum. Same way they also have a cis phase and a trans phase. In this cis phase and the trans phase there are a lot of vesicles. The main function of Golgi apparatus over here also will be same, to modify proteins, to modify fats and then release. Also in an animal cell, Golgi apparatus is involved in communication between the two cells. Cytoplasm. In an animal cell, the cytoplasm will be same way watery fluid kind of a matrix. Now this matrix, there are some places in the cytoplasm where there are no cell organelles. So such a place where there are no cell organelles, we call that place as the cytosol of the cell. So cytosol is basically a place where there are no cell organelles. The next part which we will be looking forward is vacuole. Now if you see a vacuole is a cell organelle in an animal cell which is very small as compared to the one which we had seen in plant cell. In plant cell, the major part of the cell was occupied by this cell organelle. But in an animal cell, it is a very small organelle. Also, in animal cells, it is temporary vacuole. It is not a permanent vacuole. The next thing which we have to see is about the nucleus. Same way nucleus, which has chromatin fibers, it has nucleolus in it. The nucleolus same way will help the nucleus to maintain its proteins. Also, the chromatin fibers during cell division will come together, they will form chromosomes, and we know chromosomes are made up of the molecule DNA, deoxyribonucleic acid. This is the main molecule which will pass on all the hereditary information from one generation to another generation. The next cell organelle which is there is mitochondria. Same way, mitochondria is double membrane. So the outer membrane and the inner membrane. The inner membrane will be into folds. These folds we call as cristae. These folds are the place where the cell will take all its metabolic activities and the energy-rich molecule which we call as ATP is generated. The next thing which we will be looking forward today, in an animal cell, there is no plastid. So an animal cell will lack plastids and it will also lack a cell wall. And that is the reason animal cells will not have a proper rigid shape like the plants. The next cell organelle which we have to look are exactly above the nucleus. We have one cell organelle which is called as the centrioles. These centrioles, they are always 90 degree with the nucleus. The main function of this centriole is during cell division. When the cell division in an animal cell is initiated, these centrioles divide into two and they move towards the opposite poles and they make something called as the spindle fibers. So they make up these spindle fibers and these spindle fibers help in the cell division process. Okay students, now we move forward to study something more about the centrioles. So centrioles, as you see, they are placed above the nucleus. These cell organelles are exclusively found only in the animal cell. You will not find these centrioles in the plant cell. So first let's understand its basic function. So the basic function of a centriole is to help the cell to divide. How it will do so? So when the cell division will start, these centrioles, they divide themselves, move towards the opposite poles of a cell and then they release something called as spindle fibers. So once these spindle fibers are released, these spindle fibers help the cell to divide. Now if we have a look at the structure of these centrioles, they are made up of something called as microtubules. So if you have a look at the structure of the centrioles, they look like this. And the microtubules which are present, these microtubules are arranged in 9+2 fashion. 9+2 means 9 microtubules are together plus 2 microtubules. And the two centrioles, they are always in pairs and they will be perpendicular to each other. And once they are perpendicular, they will divide during cell division and release spindle fibers. Now let's have a look at the next cell organelle, which is the cilia and flagella. Cilia and flagella we have already seen in a bacterial cell, but there are some cells, animal cells, who have cilia and flagella. So these cilia and flagella, they are the extensions of the cell membrane that are lined with the cytoskeleton. So in the flagella, they are all lined up with cytoskeleton. Flagella are generally much longer than the cilia and they are also whip-like. But there are often hundreds more cilia than flagella on a given cell. So flagella, they are primarily responsible for the cell movement. How a cell moves is because of flagella. The flagella and cilia, they are covered with a plasma membrane. So if you have a look at the structure, this is how cilia and flagella look. They have a plasma membrane as well. The core is called as axoneme. They possess a number of microtubules which are running parallel to the long axis. So just have a look. This is the microtubule which is present and this is how they are all running parallel to it. The axoneme usually has 9 pairs of doublets which are arranged radially to the peripheral microtubules. Also, it will have a central microtubule arranged to it. Such an arrangement of axonemal microtubules is referred to as 9+2 arrangement, which we have already seen in the centrosome. And they are also having one bridge which is also enclosed by a central sheath. And this central sheath is connected further to tubules which are peripherally doublet by a radial spoke. So this is how the entire structure of the flagella and cilia looks like. It also has a dynein arm which is present. This dynein arm will help in the movement of this cilia and flagella and the cell will be able to propel itself ahead. This is all we have to study in this chapter. So we have studied about the plant cell and its cell organelles, we have studied about the animal cell and its organelles, and we have also covered with the bacterial cell. Thank you.